So welcome to our second research conversation of the semester. Thank you everyone for joining. Today we are fortunate to have Shikha Prashad here, who is an assistant professor in kinesiology at the WSU Pullman campus. She specializes in studying brain networks that underlie our motor behavior in typical and clinical populations across the lifespan. This might include movement disorders and substance abuse. And she primarily uses behavioral methods and EEGs to examine patterns in behavior and brain activity and how those are disrupted with age and different disorders. And today she's going to talk to us about learning motor skills, insights from Parkinson's disease, and cannabis use. We will be recording today, so after it's finished, we'll post this on the college website within a week for people to view as well. And our format, again, for our research conversations is to have our presenter talk for around 20 to 25 minutes and then really have a conversation around the work that's happening and open up for questions. So Shikha, I'll turn it over to you at this point. And thank you for taking time today to present to us.
Of course. See if I can share my screen. Is that good? Or are you seeing the presenter view? Okay, perfect. Great. So thank you, Brian, Laura, and AG for inviting me to share some of my work. And thank you to all of you for attending yet another event on Zoom. I know that everyone's busy and tired, and time and mental resources are really precious these days. So I appreciate your time and attention. So I direct the Cognitive Motor Neuroscience Lab in the Kinesiology program at WSU in Pullman. And today I want to share some of my work on how changes in dopamine levels in the brain affect motor learning. So I actually did this work during my PhD at the University of Maryland in College Park and as a postdoc at the University of Texas at Dallas. I joined WSU last year and I got a lot of great advice about being new faculty, but no one told me that I have to navigate a pandemic in my first year along with everything else. So I'm not quite yet ready to share anything new yet, but hopefully soon. So I actually want to start by talking about a case of a patient with Parkinson's disease. Parkinson's is the second most common age-related neurodegenerative disease after Alzheimer's. And it's caused by the degeneration of neurons in a specific area of the brain that are regulated by the neurotransmitter dopamine. And so a treatment is to give patients medication that bind to dopamine receptors. So this patient was prescribed a dopamine medication to control his motor symptoms. And after about a year, he began to notice something really strange. He started to gamble almost daily, and he lost like $3,000 over a few months. And previously, he had only gambled once every 4 to 5 years, and he spent like $20 per visit. So this was a huge change for him. And he brought this up with his neurologist, and his neurologist immediately started tapering off his medication. And within a month, the patient reported no more interest in gambling. So that's really strange, right? Well, it turns out that pathological gambling is actually a potential side effect of treatment of Parkinson's disease, and it's related to increased levels of dopamine. And this is because dopamine is not only important for movement, which is why the symptoms of Parkinson's are primarily motor symptoms, but dopamine is also important for experiencing reward. And so it acts as a learning signal that shapes behavior. And so changes in dopamine levels can affect how we maximize rewards and avoid punishments. And these levels can cause impulse control disorders as well as substance use problems. So I got interested in Parkinson's disease and later substance use through my work in motor learning. Motor learning is a fundamental process for performing all of the complex motor behaviors that we do and that emerge from simpler movements that we produce in a specific order. And knowing this order is really important. So it's critical for writing, for playing instruments, for driving, for playing sports and maintaining a healthy lifestyle. It's important in development, in learning new technologies and tools throughout our lifespan. we've all recently had to adapt to new technologies. We've had to learn Zoom and other tools for our classes. And it's also critical for recovering from injuries or disease. So it's really important for us to be able to adapt to changing factors, for example, in the environment, like being able to drive in different weather conditions, or in the tools and technologies that we're using, or just changes in our own body that are occurring due to aging, injury, or disease. So as an example, this is Sven from the movie Frozen. And now Sven is a reindeer, and so he can easily gallop over the snow, and he can even pull a sled with Kristoff, Anna, and Olaf in it. However, on ice, Sven isn't so successful, and he can't just gallop over the ice. But because Sven has intact motor learning, he quickly adapts, and he determines that the best way for him to move across the ice is to do this. And so motor learning is not only important for activities that are essential for daily living, but it's also important for activities that bring us meaning and joy through sports and hobbies and communicating with people and allow us to do our work. When thinking about how movement is controlled in the brain, there are a lot of cortical structures, which just means areas on the surface of the brain that are critical, like the primary motor cortex, which is a strip that goes from one ear to the other. And the primary motor cortex is involved in executing movement, so it sends signals to our muscles to initiate movement. The premotor cortex and the supplementary motor area help organize movement into sequences, and the prefrontal cortex helps plan movements, and it's also a critical region for cognitive function like attention and decision making. And these areas are also served by other sensory areas that are receiving sensory information from the environment. Along with these cortical regions, there are subcortical regions, which are regions below the cortex that are critical for movement. Of these, the most important and my favorite are a group of structures called the basal ganglia. The basal ganglia have vast connections with pretty much the entire cortex, but they include the frontal areas and the motor areas. And so it's involved with controlled movement and cognition. Parkinson's disease is caused by the degeneration of neurons in a particular structure within the basal ganglia called the substantia nigra, which is right here. And the substantia nigra supplies the dopamine that regulates movement pathways that these different structures are a part of. Some of these pathways facilitate movement, others inhibit movement. And by modulating these pathways, dopamine facilitates voluntary movements and allow us to move when we want. In Parkinson's disease, a depletion of dopamine causes these pathways to become imbalanced, and the net result is that there is a general paucity of movement. And so patients will have difficulty in initiating movement. They'll exhibit slow movement, tremor, rigidity, postural instability, and some will even have diminished facial features, and they will blink less. So let's look at the changes in how dopamine can affect behavior. So in a typical adult, the levels of dopamine throughout the brain are at their optimal level, and that optimal level can be different for different regions. So as an example, there are 4 regions in the brain. The dorsal and ventral caudate are part of the basal ganglia. It's this purple structure. The nucleus accumbens is this yellow dot here. It's part of the reward system that underlies substance use, and the frontal regions underlie cognition. So these are just these frontal regions in general. So in a typical adult, these levels are at their optimal level. In patients with Parkinson's, the dopamine levels in one part of the basal ganglia, the dorsal caudate, decrease, but the levels of dopamine in the other regions remain optimal because the loss of dopamine is very localized and it happens to be at the location that's critical for movement. And here off means that the patients are off medication. So they're not, these are their levels when they're not taking medication. Now, when patients with Parkinson's take the dopamine medication, it reduces the motor symptoms because it restores the dopamine levels in the dorsal caudate. But because the medication is not targeted, it increases dopamine levels throughout the brain. And so dopamine levels increase in the other brain regions. And this means that there is a lot more dopamine in the nucleus accumbens, which is part of the reward pathway that underlies substance use. And actually, a hallmark of substance use is increased dopamine in reward areas, and that produces the effects of many substances and causes individuals to become dependent on them. And dopamine levels in the frontal regions also increase. And in terms of cognitive function, the relationship between cognitive function and dopamine levels is an inverted U shape. So too little and too much dopamine both impair performance. And so increased dopamine in the frontal regions can lead to impaired cognitive function as well. So while Parkinson's disease has been characterized as a movement disorder, there have been inconsistent results regarding the effect of Parkinson's on the learning of new motor skills. Some studies have shown that patients with Parkinson's exhibit impairment in motor learning, but others have shown no impairment. And it's also been suggested that impairment may be a function of disease severity. Treatment like dopamine medication and deep brain stimulation surgery, which is done in later stages when dopamine medication is no longer effective, may have differential effects on learning and brain activity as well. Now, we also have to remember that typically patients with Parkinson's aren't just affected by Parkinson's, they're also undergoing aging. And the consensus is that typically aging adults can perform at similar levels as young adults if the sequence that's used in the task, and I'll get to that in the next slide, is not complex. And the decrease in performance for more complex tasks is related to declines in working memory capacity and structural changes in the striatum, which is also a part of the basal ganglia. So the task that we used was the serial reaction time task, which is the most widely used task to assess motor learning. In this task, participants are presented with 4 locations on a computer screen. And at the beginning of a trial, the stimulus appears in one of these locations. For example, this one turned blue. And the participant must press the corresponding button, which could be a button box or a keyboard key, as quickly and accurately as possible. Now, participants don't know that these stimuli are actually appearing in a sequence. So if we number these locations 1, 2, 3, and 4, then this could be a sequence that the stimuli follow. And the sequence is repeated 10 times in one block. And the idea is that as participants implicitly learn that there is a sequence, their response times get faster and faster because they're able to anticipate what the next stimulus will be. So over the course of 7 blocks, this is what we expect how response times will change if the participant exhibits learning. So this is mean response time in milliseconds on the y-axis. Faster responses have smaller numbers, so a downward slope is actually better performance. It's a little bit counterintuitive. And the blocks are on the x-axis, and B0 is the baseline block. So we're just getting a sense of what their baseline response time is. And so stimuli appear in a random order in this block. In blocks 1 to 4, the stimuli appear in the sequence. And so we expect that their response time will get faster and faster. In block 5, stimuli actually appear in a random order again. So because the participants aren't able to anticipate the next stimulus anymore, we expect reaction time to significantly increase. And then finally in block 6, we show them the same sequence and we expect response time to come back down again. So this decrease in the first 4 blocks, increase in block 5, and decrease in block 6 is what we expect participants to show if they are able to successfully learn the sequence. Now, a problem with the serial reaction time task is that it equates reaction time and response time, but these are not the same. So response time is actually the sum of reaction time, which is the time it takes to react to the stimulus and initiate movement, and movement time, which is the time it takes to actually execute the movement once it's been initiated. And presumably this was because in the traditional tasks, the participants' fingers are on top of the buttons they're going to press. And so the assumption is that the movement time is negligible. But it seems strange to me that studies were assessing motor learning in patients with movement disorders like Parkinson's without taking into account that these are movement disorders. And so the task kind of overlooks the movement aspect. So he came up with a modified version of the task that uses the number keypad on the keyboard instead. So participants start with their index finger on the 5, which is in the center. So that's like their home button. And the idea is the same. One of these 8 locations, the stimulus pops up in one of these 8 locations, and then the participant presses the button that corresponds to that location. So the trial starts with the participant pressing 5. After a short interval, a stimulus appears, for example, here. Then the participant releases the 5, that's the reaction time. And then they press the corresponding key, that's the movement time. And then together we can measure response time. And so in this way, we can separate out the cognitive processes measured through reaction time and the motor processes, which are measured through movement time. So for this study, we collected data from young adults, typically aging adults, and adults with Parkinson's. And patients were in the early stages of the disease, and they had been diagnosed with Parkinson's for an average of about 6 years, and they were all tested on medication. So this is the same plot as the one I went over before. The mean response time is on the y-axis, and the blocks are on the x-axis. And in the young adults, we saw the same pattern of decreasing response time in blocks 1 to 4, increasing response time in block 5, where the stimuli were in a random order, and decreasing response time in block 6 again, where the stimuli followed the sequence. Now, since we use the modified serial reaction time task, we were able to separate these into reaction and movement time. And we found that for reaction time, it followed the same trend with a decrease, increase, and decrease, while the movement time was pretty constant throughout the task, which is what we expected. We do see a little decrease in response time in the first block as participants get familiar with the keypad and are able to move faster, but after that, it stays constant. In typically aging adults, we saw the same pattern. The only difference was that they were a bit slower overall compared to young adults, and that's expected. When we decomposed it into reaction time, we saw the same pattern in reaction time and constant movement time as well. So let's take a look at the patients with Parkinson's. So immediately you can see that the patients with Parkinson's were different from the typically aging adults. So one is that they don't show the pattern. In fact, they're fairly constant throughout. And 2, look at these huge error bars. There's so much variability in that group. And that's actually a characteristic of Parkinson's, that it's very variable. Some patients progress through the stages very quickly, and others spend 10 or 15 years in the early stages and they don't get worse. And so the symptoms that they experience can also be different. And so this actually makes Parkinson's very difficult to diagnose. And in fact, by the time patients are diagnosed, they've lost about 60 to 80% of the neurons. And so there's a really tiny window for the treatments to work. And so if we can diagnose Parkinson's earlier, we can enlarge that window and develop better treatment and maybe even a cure. So this was response time. And when we decompose this into reaction time, we found something totally different. So the individuals with Parkinson's actually did show the pattern. They decreased their reaction time in blocks 1 to 4, increased in block 5, and decreased again in block 6. So that means that all of this variability is coming from movement time. And that's also makes sense because Parkinson's is a movement disorder, and one of the hallmarks is slow movement. And so it makes sense that their movement times were slower and more variable. I honestly was really surprised when I saw this result. I didn't expect to see such stark differences between reaction time and movement time. And I fully expected it to just be a mix of both, where both just show slower and constant reaction and movement times. But I was really surprised to see that there was this really interesting distinction. And so this suggests that patients with Parkinson's might actually be able to learn motor skills, at least in the early stages, and that it's really critical in these populations to separate the cognitive and motor deficits. So I was really excited about this result. And when I got to my postdoc lab, which studies the effects of cannabis on the brain, I was excited to explore whether cannabis users, which as we discussed earlier, can also experience changes in dopamine levels, also have impaired motor learning. And I was really surprised that there were no studies that assess the effects of cannabis on movement. Most studies focus on the effects of acute cannabis on driving, which is understandable, but none investigate how processes underlying skills like driving are affected by chronic cannabis use. And so we collected some pilot data using the traditional serial reaction time task because we wanted to be able to compare to other populations since there were no studies in cannabis users yet. And because this was in the modified version, we weren't able to decompose the reaction time and movement time. We collected data from 10 cannabis users and 10 non-using controls. And these participants used cannabis recreationally about 3 to 4 times a week on average. And most didn't meet the criteria for cannabis abuse and dependence. They were relatively light users. And this is the same plot, mean response time on the y-axis and the blocks on the x-axis. The blue are the non-using controls, and we can see that they follow the pattern with the significant decrease, increase in block 5 and decrease again in block 6 as expected. The cannabis users shown in green didn't show this pattern, and their response time stayed pretty constant throughout. Suggesting that they weren't able to learn the sequence. So this is different from the patients with Parkinson's, right? There's something different about the way that dopamine interacts with motor and cognitive systems in cannabis use. That's different from Parkinson's disease, but what that is very unclear. So based on this preliminary data, I wrote AR21 grant to look at this further. That was funded, and so we're currently collecting data, so hopefully you'll have more results to share. And that study does use the modified serial reaction time task. We'll be able to separate the reaction time and movement time. So we did measure brain activity during rest in a larger group of cannabis users. They had the same demographics as the ones that did the motor learning task. And resting state is when participants are not engaged in a task, and it reflects the baseline functional organization of the brain. And so it's an important indicator of how robust the networks that underlie cognition are, and possibly the networks that underlie motor processing as well. And we measured brain activity using EEG, which is a brain imaging technique that measures electrical signals that were always just emanating from our scalp, using electrodes that are fitted on a cap that kind of looks like a swimming cap. And one way to analyze EEG data is to decompose the EEG signal into different frequencies that correspond to different cortical processes. So for example, delta frequencies are large amplitude slow waves. They're more prominent during sleep, but are present during wakefulness as well. And beta are seen when we are alert and engaged in a task. And these are topographical maps. I don't know if you can see this, but there are these tiny dots on each of them. And those represent each electrode, and the red represents greater cortical activity and the blue represents low cortical activity. And this row shows the cortical activity in non-using controls. And so during rest, when participants are not engaged in a task, we expect more activity in the delta bands and low activity in beta, like we found here. But in the cannabis users, we found the exact opposite. So they had significantly decreased power in the delta band and significantly increased power in the beta band. And this suggests that cannabis users had greater cortical activity even during rest, which looked like they were actually engaged in a task, which could mean that they weren't able to inhibit their ongoing neural activity. And this extra neural noise may affect networks that underlie cognition and perhaps motor processes, and that could explain impairments in cognition and motor learning. So in this study, we didn't measure cognition in these participants, so we weren't able to correlate performance in cognitive studies to increased cortical activity. But that's the next step. I actually received a pilot grant from the alcohol and drug abuse research program here at WSU earlier this year to further investigate whether these changes in resting state are associated with cognition. So my aim is to continue examining these interactions between cognitive and motor processes in both typical and clinical populations, and trying to disentangle their contribution to motor learning. And I just want to make a quick plug for trying to understand movement. I feel like motor processing and movement is often overlooked and undervalued in areas when studying things like cognition or learning, but it's an integral link between brain and behavior, because all of the behavioral outputs that we measure, which could be reaction time through button pressing, maybe you're asking participants questions, that speech, these are all motor skills and they're modulated by motor processes. And so that makes movement and motor processing our primary gateway to understanding cognition and behavior and learning. And so I invite you to think about the role of movement and motor processing in your own work. And you might find that it plays an important role in the interpretation of your data, or maybe even the way that data is collected. And I also am interested in understanding the patterns of neural disruption that may underlie these disorders and contribute to the cognitive or motor impairment. It's really important to characterize individual differences because they can influence how we interpret data, and often individual differences are overlooked when we compare averages across groups. And I didn't have time to go into this today, but I also looked at the group of typically aging adults in the motor learning study. And I found that there actually was a subgroup in there that showed impaired motor learning. So they actually performed worse than the patients with Parkinson's. And I discovered this using a machine learning algorithm called k-means clustering that identifies subgroups within a data set without any predetermined criteria. So the algorithm doesn't take into account their ages, just their performance on the serial reaction time task. But this information is completely washed out when we're just comparing averages between groups. And so age or presence of Parkinson's disease or use of cannabis are just individual factors that are related to impairment. They're not the sole causes. And so they may affect individuals to varying degrees. And we already know that in aging, someone can be 75-year-old, but be more highly functioning than someone else who is 60. And we know that the symptoms of progression of Parkinson's varies widely between individuals. And some people can use cannabis a lot and still maintain high level of functioning while others can't. And so all of these factors and other factors and their interactions can get really messy. And I think that applying computational approaches may be one way to disentangle some of these systematically. And I just want to end by quickly acknowledging just a tiny slice of people who have made this work possible. My PhD and postdoc advisors and labs and the participants for letting me take a peek into their brains, and the funding agencies, and to all of you for your attention.
Thank you, Shikha. That was very informative and very interesting. So let's open up for questions and conversations. And I could start, but I won't. I'll let other people start. I'll fill in.
Can I start?
Sure.
All right. I'm thinking about your anecdote at the very beginning about the gambling issue, and I'm looking at your serial reaction time and how you can implicitly, or you're expecting people to implicitly learn the process. And I'm thinking this is a lot like gambling. This is essentially what gamblers are thinking, that they can count the cards, or they'll learn, or they're pulling the slot machine. I know it's going to hit on the next time. So I'm thinking, what is? I'm wondering what is how are Parkinson's individuals doing on something completely random? And going back to that slide, I don't know if you can show that slide again, but the Parkinson's individuals on that, you mentioned that it was related to the medication, and were those individuals on the medication versus off the medication? I guess you maybe can talk a little bit more about that relationship between that antidote and that task for me. Thanks.
Yeah. So the participants were all on medication when we tested them, and we timed the study such that they were at approximately like 1.5 hours after they had taken the medication. So approximately at about the same stage. And I think your other comment about the implicit learning and gambling being similar is really interesting. I've never thought about it that way. But I think one thing that's different is that gambling has a lot of risk. And so that's what keeps people going, right? Because you don't exactly know what's going to happen the next one. You think maybe the next one will be the one that will make me the money. And when it does, that's when you have that dopamine spike, and that feels good. And then you want to have that spike again. And so you keep going. Whereas in the serial reaction time task, when participants are doing that task, they're literally just told that it's a reaction time task. You're pressing buttons as quickly as possible, and we're just looking at how fast you're going. And so most of them have no idea that they are learning anything. And even at the end, we ask them whether you think that there was a, something going on with the locations, and they usually say that it was totally random. When we show them snippets or chunks from the sequence, they aren't able to recognize them as what they had been doing. So for them, it is just a reaction time task, and they don't even realize that they have been learning a sequence.
Since we're talking about reaction time, can I ask a methodological question?
Mm-hmm.
So I really appreciate the detail of parsing out movement versus response or reactions. And I'm curious from your perspective, if you could have other measurements that might even be more sensitive to the cognitive processes. And I'm thinking of how we use like eye tracking behavior or eye tracking measures to get at some of the cognitive processing we think is happening. And if you were to use something like that with patients in Parkinson's, would you see the same type of trend, just different reaction times? Would that be a more sensitive measure to not the movement, but more the cognitive component of this? And have people tried that?
Yeah, I think adding something like eye tracking would be really interesting in this task, especially with patients with Parkinson's. Um, I don't think that's been done, at least in this group yet. Another way that we actually measured cognition was we did actually have them wearing an EEG cap, so we do have their brain data as well. We have their resting state and while they're doing the task. And one of the I think most important things that I found in that was that even though the patients with Parkinson's were performing at a similar level behaviorally as the typically aging adults, they required greater cortical activation to attain that. So we saw while they were doing the task, there was more cortical activity compared to the typically aging adults. So other studies have also found that might be a way as a compensatory mechanism that the brain works harder to get the same behavioral result. And that's not always, you can't see that when you're just looking at behavior.
Thank you.
I have a question. I've always wanted to ask, and I've never asked you, but could you explain more about the machine learning techniques and like what is that exactly? And so I found that so interesting, the ability to identify these different kind of profiles or patterns at maybe a more fine-grained level is how I'm perceiving how that works. But and able to identify where there's individual difference factors that maybe weren't known before. Could you just explain a little bit more about what that is and how it's done?
Yeah, so I actually ran that as just an exploratory thing, just to try it out. I wasn't really expecting to find anything. And I basically just put in all of their individual reaction times for every single trial throughout the entire block for all of the typically aging adults and the patients with Parkinson's into the algorithm. And what the algorithm does is that it takes all of that information and it just tries to figure out how to group it such that There are participants that are more close to each other are formed into one group, and then those that are far away are formed into their own group. And so actually I can show you, I think I have some slides. I always have some extra slides for questions afterwards. Let me just see if I can find them. And we found these different, here it is.
So while you're looking, I just want to, so this sounds a lot like something that we do. It's not technically statistics, right? With cluster analysis or latent profile modeling, where you're, again, looking for or identifying clusters or patterns or profiles based on maximizing similarity between the groups. Maximizing similarity and maximizing distance between those groups. So I guess I'm just kind of curious how that is maybe different, or if you're familiar with either of those other analytical approaches for profiling.
Yeah, I think they're actually very similar. I mean, machine learning is basically stats. It's just a fancy way of saying stats.
That's helpful.
Yeah.
Cool.
Yeah, I mean, all of, machine learning, they use things like words like features, that just means variables, independent variables that you're including. So it's exactly the same. Even like regression is a machine learning approach. So yeah, it's exactly the same. It works in exactly the same way. So this is actually what we found. So it separated the typically aging adults into these different clusters. I have working memory on here now, but when it did this, I didn't know what it was that was separating them. And just by looking at their data, I realized that it was actually separating them by working memory. But working memory wasn't a variable that was inputted into the algorithm. The only thing that it had was the reaction times. But it separated them into these groups based on their working memory capacity, and the participants that were impaired at learning had the lowest working memory scores. And when we looked at their age, there was no difference in how old they were between the different groups, but there was a difference in the working memory capacity between the highest performing group and the lowest performing group.
And are those differences identified sort of post hoc then? So these groups are formed based on reaction time, and then you explore how those groups are different. Is that correct?
Yeah. Yes.
Okay. Huh. Do you know if you can take into consideration multiple features or variables in creating those profiles or clusters?
Yeah, so you can enter as many variables as you want into the algorithm, and it will figure out what if there are any subgroups and what those are.
Cool.
And I'm just thinking on the same lines, like methodologically, there's a lot of work being done in terms of intensive longitudinal data modeling, which is essentially what you have with these reaction times happening all at the same time. And people do the same thing when they collect like ecological momentary assessment data, where they're asking someone questions, every hour or every few hours. And some of the modeling techniques, I'm wondering if they would apply here, because they do allow you to see variability at the very individual level and not just the group level. So it's almost one step above doing like a growth curve model where you can see individual variability as well. But this is really looking at the individual level. And I know they're doing this in some educational data now, both in kind of latent or structural equation modeling, as well as observed score models. I'm just thinking there could be a connection with some of those models to what you're trying to look at, and if that would allow you to see the same patterns, or if it would result in finding something you haven't seen yet.
Yeah, I think that's really interesting, and that is something I want to look into further, because I think, Jeffrey Herring at Maryland, he was on my dissertation committee, and he said exactly the same thing when I showed him this data. And so, yeah, so that's been something that's in the back of my mind that I want to explore with this data.
Okay. And Jeff's great, very knowledgeable person. Yeah. Well, there's a new article, maybe I'll send to you in Psych Methods that just came out that talks, it's a primer on some of these methods. So I'm happy to send that to you, and has some good references in there for some of the people who have done some of this work outside of education as well.
Yeah, that would be great.
Okay.
Yeah, thank you.
So let me follow up one more thing is what other individual difference factors or variables do you think would be important to explore?
Yeah, so I think that a lot, because this task it's not just a motor task, it's primarily actually a cognitive task. I think there are a lot of cognitive variables like working memory that are different among individuals. Other things like attention, because if participants aren't paying attention to the task, then they're not going to learn the sequence. And we've also seen that participants who don't pay attention, you can have you can measure attention through EEG as well. When we've seen that when they aren't paying attention, their response times don't improve and they pretty much stay flat. So that's another one. There are a whole host of other cognitive functions that could be different. And I've also used this computational approach to look at neural response and behavioral response in heavy cannabis users. And we specifically wanted to look at sex-related differences, whether men and women respond differently to the effects of cannabis use. So we looked at their brain response when they're looking at cannabis cues, which usually triggers craving in these very heavy cannabis users. So these are users who use recreationally. They've been using, they have a minimum of 5,000 uses in their lifetime. So if they're using once a day, that's 365 in a year. So they have to be using like for 10 years about. And I think the average in that group was 14 years they had been using. So they're very heavy users. Looking at marijuana cues is very triggering for them. And it kicks in their craving response. And so we looked at how they respond using fMRI. So looking at how their different brain regions responded to cannabis cues. And then we also asked them their subjective, like how much are you craving cannabis right now? And we found that we used PCA to combine the neural and behavioral responses and to see which one described more of the variance in the data. And we found that the neural response was the most, the variable that accounted for most of the variance in the data set, but there weren't any sex-related differences. But subjective craving, which was the second most, accounted for the second most variance in the data, that did show differences where women reported greater subjective craving compared to men. And that suggested that there's something happening between the neural response to how you feel subjectively, something happening, probably some interaction with hormones that has a very different effect in women. And that might explain why a lot of treatments and interventions have poor clinical outcomes for women, and women don't get as much out of them. And that might be because the approach doesn't take into account that they're actually subjectively feeling very differently when they use cannabis. So that's another example of individual differences using machine learning.
Other questions or comments?
Can I just ask too about, so the grant that you applied for, the WSU one, it's like an alcohol and substance use. Is that focused only on cannabis, or are you also, or have you looked at other types of substance use and look at similarities or differences? With either, well, I had two questions. One was about other substance use patterns, if they're similar, as well as other patterns of things like addictive type behavior. So you talked about gambling, but there's lots of addictive type behavior. So those were two different areas that I thought were really interesting to see if they are similar or different from what you're finding with with the cannabis in Parkinson's patients.
Mm-hmm. Yeah, so that grant is just looking at cannabis users, and it's just specifically trying to see if there is a relationship between resting state and cognition in heavy cannabis users. I haven't looked at other substance use. In my postdoc lab, we did a little bit with nicotine, and we actually looked at resting state in individuals who only use nicotine, and then those who only used cannabis, and then those who used both. And we actually found that there were, if you only use nicotine or cannabis, your resting state activity looks very different from controls who don't use any of these substances. But if you're using both, then it looks very similar to controls. So our conclusion was, if you want to use both, you know. But yeah, so I haven't really looked at alcohol or any other substance. And I think that looking at behavioral addictions is also really interesting, because there isn't a chemical that's, you know, or external substance that you're taking that's affecting your neural response or binding to receptors in your brain. And so yeah, I find those really interesting, but I haven't looked at those yet.
Okay, so the first thing that came to mind is I was thinking about people who are like addicted to exercise.
Mm-hmm.
Like, what would happen with that group in particular? Because then you have sort of the exercise movement component as well. So that could be interesting.
Yeah.
Shikha,
Yes.
very nice presentation. Thank you so very much.
Thank you.
I don't think well, this might not be a question, but something that you probably might want to consider, or maybe you have considered this. I came into your presentation a few minutes late because of another appointment. Listening to your talk, I could see a lot of conceptual and methodological advancements and the possibility of connection with so many different units on campus. So I don't know if you are giving some thoughts into how you can connect this work with the broader research enterprise on campus, in medical school, in psychology. Brian talked about, you know, a new method. I know Brian, Dai, and Ola did some work about that method last year. Brian, please correct me. So I don't know if that's something that you are thinking short term to connect this work to many other research activities going on campus.
Yeah, definitely. There are a lot of people on campus that are doing either something similar or something complementary. So yeah, that's definitely something that I am interested in seeking out those people, and Brian has helped with making some of those connections already. So yeah, and I'm going to continue doing that.
Thank you.
Yeah, I mean, one other thought too that, you know, is when you were talking about kind of like the learning component, I was kind of curious, which you said is really implicit learning, but I was trying to think of applications with, you know, education, like more explicit learning.
Mm-hmm.
But you kind of explained that with Robert's question about kind of that it's not very explicit.
Yeah, so you can actually do this task with an explicit component where you tell them that there is a sequence, and that can be done in different ways. So there's one way where you tell them, okay, there's a sequence, I want you to look for it, and I'll ask you what it is afterwards. And then a lot of studies have actually found that telling them that there is a sequence and to look for it, it actually makes their learning worse, and they aren't able to find it because that
Trying
explicit
too hard.
yeah, exactly. They're trying too hard, and that blocks the implicit learning. And then the other way is to just give them a much more simpler sequence. So not, I think this was what, 12 items. So maybe it's like 5 or 6 items, and you have them practice it before they start the task. And that's really a more motor task because there really is no learning aspect.
Yeah, makes sense. And I think we've talked before, is it, with something like ADHD, does that fall under the same kind of dopamine dysregulation category?
Mm-hmm.
Am I right about that? Okay, so yeah, so that would be really interesting to also to look, at that group. And
Mm-hmm.
I know there's, I know some people who are doing some kind of exercise or activity interventions to help, ADHD students in schools to be more successful. Yeah, I guess one final question that I have is your thoughts on using exercise. You talked about, so at the very beginning, about how the effective medication in Parkinson's patients actually then increase dopamine too much, and that
Mm-hmm.
isn't good. So my first question was, wow, what if exercise is thought to increase dopamine, so should we not have certain people exercise? Or what is the thought on using exercise as a treatment or therapy, or just being recommended?
Yeah, I think that's a really important point. And I think that the way that dopamine is involved in so many different aspects of behavior, and it's everywhere. All the brain regions are involved. So the way that, like Parkinson's or exercise affects dopamine levels, and like the downstream effects can be very different.
Mm-hmm.
So a lot of studies have found that if you do exercise before doing this more learning task, then you perform better at the task and you learn faster, because exercise also improves other things, like, right, improves plasticity, and all of that has a positive effect on learning. So yeah, if anything, these studies suggest that you should exercise more, especially just before you're going to class or taking a test, and that should increase performance.
Yeah, well, that was my first thought. And then I thought, wait a minute. Oh, no. But what about, I wonder maybe that, okay, I got to stop talking, but what about the interaction between like like for a Parkinson patient in particular, like what if you exercised with the medication? Would exercise help regulate better because of these other, like you said, downstream effects?
Yeah, I don't think studies have looked at how exercise and dopamine medication interacts in patients, but there have been a lot of positive effects on their motor symptoms if they exercise. And so for, at least for the motor symptoms, exercise is beneficial for patients as well.
Okay, phew, good. Right, thank you so much. I appreciated your talk a lot, and you answering all of my questions and giving me some more insight into what you do. So thank you.
Of course, thank you.
Yeah, Shikha, I would echo that. Thank you very much for presenting today. Very informative. I really enjoyed the way you make some complex things you're doing very understandable to us. So thank you for that. I know I have some questions. I don't know how, I have to think about how I'm going to phrase them, because they do go along with some work that Bruce and I are doing with Oregon Health Sciences, looking at motor and cognitive decline in rare neurological and muscular diseases. So there's something there. I just don't know what it is yet. When we have data, I probably will connect with you to talk about that a bit. But I really enjoyed your talk, hearing about this, and I really appreciate you taking time to do this. I will remind everyone we have our third talk for the semester on December 3rd. That'll be at Sports Management with Scott. So look for that. Announcements will come out about that as well. So I think we're pretty much at the end of the hour. But thank you again. It's super interesting work, and I look forward to seeing it progress.
Thank you. Thank you, everyone.
Okay. See everyone later.